Good morning, everyone. My name is Jorge Mojica-Santiago. I'm a postdoctoral associate with the University of Florida in Gainesville. And I'm going to be presenting today on a microphysiological system that we have developed to test anti-atrophic potential using electrical pulse stimulation. But first, before I jump into the science, I wanted to acknowledge the pretty large collaboration that we have among interdisciplinary scientists. Dr. Shaban Malini, who is the PI of this grant, put together this massive group of bioengineers, clinician scientists, aerospace engineers. And we are looking into doing a skeletal muscle microphysiological system. Essentially, muscle itself is a complex tissue that is very dynamic and active. We know that skeletal muscle cell is a form of satellite cells that fuse into myotubes. Here we can see in red, these myotubes are fused in multiple nuclei in blue. Age-related muscle wasting, or sarcopenia in particular, is characterized by the gradual atrophy of the muscle. And so the shape that we see here gets changed over time, but it also causes a dramatic decline in mobility on an increasingly aging population. Unfortunately, because the accumulation of these changes do take time to occur, we do not have effective therapeutics to really address the problem of sarcopenia at this time. Here we can see an example, let me see if that plays a of a young, healthy muscle thigh against an older sarcopenic patient. You can see an accumulation of the fatty tissue, and so and a gradual atrophy of the myobundles or the fused myotubes. Now, what's interesting is that astronauts have for decades shown similar changes in skeletal muscle atrophy, and we thought that microgravity could help us as a model or an accelerator model for degeneration of muscle. So in fact, we are interested in understanding how human muscle biology in microgravity relates as for aging. As we all know here in this conference, microphysiological systems are great models for helping understanding human diseases in vitro. Microphysiological systems for skeletal muscle have been traditionally used in a couple decades using these two, in general, is using tensile, applying a tension on the skeletal muscle cells and in a 3D hydrogel matrix. Some folks have used photopattern hydrogels. Others have used general bundled frames that are deformable or fixed. And many have used microfluidic channels to also allow for perfusion, continuous perfusion of the skeletal muscle tissue. Now, as I mentioned before, we are interested in studying how microphysiological systems in microgravity, we are looking at how in microgravity we can model the aging of the skeletal muscle itself. Therefore, we are we're developing a platform that we can indeed look at in microgravity, how the contraction parameters change, and also eventually look at the transcriptomics changes as it returns to ground. So how we did this is we first started looking at multiple cell matrix environments. We started by combining a multiple collagen fibrin blends and also comparing this against a matricial collagen formulation has been previously published. We found that laminin and HA were important components for the collagen fibrin blends to produce a general myotube distribution across the whole the skeletal muscle. And we can also see that matricial collagen, as it's shown in the literature, does appear to form this myotube fusion index. So we decided to move forward with the matricial collagen formulation, but before we added any cell types of this, we used simulations of computer fluid dynamics, as others in this conference have shown. Simulations allow us to really look at the concentration distribution across the whole chip. And so because we were looking at a rather complex geometry, we were interested in both how the volume fraction of air is moved out. So in top here, you can see hydrogel being injected through the port and then just kind of gradually filling up this chamber. Now, over time, we see that the skeletal muscle cells pull on the matricial collagen matrix and form this rather elongated bundle of myotubes. Also, we studied how much of the flow rates we needed to apply such that we not only flooded the entire system, but also allowed diffusion of the component of the media to reach the cells that were in the middle. So we found that simulations were incredibly helpful in really finding what the right parameters were for input. Now, having developed this geometry and understood really what parameters we needed, we then took satellite cells from patients. And I just want to clarify that we looked at multiple human subjects, both on older sedentary and at young active populations. But we did pull the cells together. And so all the work that we'll be showing here has been multiple combinations of multiple samples of the microphysiological system of the pooled cells together. So what we did is we first plated the cells in 2D, differentiated them, expanded them, and then we pulled together and plated them into the collagen material formulation that we developed earlier. And we applied a differentiation media and then eventually observed how the myotube fusion occurred over a span of a week. And then after 13 days, we did start electrical stimulation to apply and to the cells and look at the contractility of these cells. And we saw that older sedentary and young athletic both did appear to form the myobundles over time. But we definitely noticed that there were some differences in the morphology of these bundles, which goes to show how these cells respond and increase variability among different patients. Then again, we looked at the simulations of applied voltage to understand what electrical field were being distributed across the whole geometry, and specifically to look at what the cells were experiencing. We identified a range of multiple voltages, and we then narrowed down to a 3 volts applied to this platinum electrode located along the media channels to achieve a desired electric field intensity at the center line where the cells were encountered. We also found that this voltage was the one that allowed us to have the minimal heterogeneities in the electric field in the middle. After applying electrical stimulation, we took videos of these bundles contractions, and we were able to use digital image correlation analysis to map the horizontal and vertical displacements of the pixels within the bundle in the different areas of the bundle. Now, we define a region of interest that at least accounted for the PDMS pillar, and we map these together, all these vertical and horizontal displacements, and obtain a vectorial summation of the displacement occurring at each location of the region of interest. With this, we then applied an average of the different displacements that we have found, and we eventually plotted this signal with multiple peaks over time. Now, what we found is eventually these cells did respond to stimulation, but rather differently. And this work has already been set up for publication. What we noticed is that during the resting phase, before we applied electrical stimulation, there seems to be some rather spontaneous contractions occurring, or spontaneous peaks that we can observe, especially for both young athletic and all sedentary. And then as we applied the electrical stimulation, the young athletic cells were the only ones that really responded in a more asynchronous manner with the stimulation that we were applying, whereas the all sedentary cells they still continue to have these random peaks, random contractions over time. After recovery or after the stimulation was removed, the cells that gradually responded stop contracting in the same way. We found that also this behavior rather correlates with the way that young athletic and all sedentary fusion looks. So if you look into the fusion index, which is a metric of how many nuclei per myotube are present in the cells, you can see that the younger athletic cells were highly fused, and then also had the better response to the contraction. We then applied signal processing techniques to reveal contractile differences between these signals that we observed. I'm going to just briefly describe here that we took the mean displacement signal of the stimulation curve that we found. We also look at the average of displacement peaks and average of displacement valleys, and then normalize these against the mean displacement of the baseline phase. We also applied a fast Fourier transform analysis to obtain the average contraction rate of our bundle. In summary, we really looked at how young athletic stimulated ships had higher frequency of contraction than the all sedentary cells. And in addition, we also had differences between phases. So we were able to show that the stimulation phase were consistently higher than the baseline phase and then the recovery. We also looked at multiple, using an RNA-seq, a PCR array. We looked at expressed myogenesis and contractility genes. In specific, we see group 1 genes that were downregulated for myogenesis. This is the group 1 over here. Group 2 were actually upregulated myogenesis genes. So it's this rather range of genes over here. And then group 3, 4A and 4B were all focused on the contractility aspects. We saw that group 3 were mostly genes that were downregulated for contractility, and 4A and 4B were upregulated. By a 1.5 to 2-fold specific tight intrapointed family of genes with electrical stimulation were found to be significantly regulated. And then since aging and increased physiological capacity are counteracting and have been shown to counteract in opposite directions in previous studies, we sought to evaluate how changing the increased the electrical stimulation regime that we applied would hopefully counteract aging in effect. So we then continue to look at these all sedentary young athletic cell types populations, but now we have applied a different electrical stimulation regime over time. So instead of just doing the 1 pulse stimulation per day, we apply 2 stimulations per day. And what we found is that, what we found is that in general, by just sheer increase in the number of stimulations per day, we obtain a higher mean displacement, signal than the 1 stimulated per day. But we also had a more consistent frequency. So remember that the older sedentary cells were a little bit asynchronous, and they had more spikes all over. We were able to sort of rescue the older sedentary to have a more synchronous response to the young athletic by just sheer exercising these cells. Now, not only are we interested in looking at the contractility aspect, but also what is it a way that we can use the system to test for drug screenings. And so we're looking at contractility recovery verified with timatidine, which is an anti-atrophic compound that is obtained from green tomatoes. So timatidine has been shown to is thought to increase the protein synthesis, mitochondrial DNA, myotube size, the expression of anabolic gene, and also cause cellular hypertrophy. We hypothesized that by adding timatidine into a skeletal muscle system, we could look into increasing muscle mass and strength, and also improve the exercise capacity. So to really validate this, we combined both the aspect of all sedentary cells and young athletic cells being exercised twice per day with different doses of tomatidine. So specifically, we tested a 10 micromole in the dosing of the tomatidine against a 5 micromole. And we all compared this against a control that was ethanol dose only, so no tomatidine added. What we found is, in general, the dominant frequency of the myobundle from these older sedentary cells were, again, synchronized with the young athletic-derived myobundles, like we had seen in the past, with just twice stimulation per day. Not only that, but the mean displacements of the young athletic continues to be higher than the older sedentary. But one really interesting fact that we weren't accounting for is that this time around, the older sedentary cells during the eastern phase appear to just continuously increase with time. So it appears as though the cells are not, the muscle bundles are not really recovering and just continue to remain in tension during the stimulation phase. That was definitely not observed when we applied the tomatidine drug treatment, both at the 5 micromole and the 10 micromole treatment. So as you can see, the blue line here, the older sedentary do not have that continuous tension increase with respect to time. As of now, this is preliminary data, so I just want to highlight that this is continuously being worked at, and we're looking at replicating these results further and hopefully obtaining more parameters like the ones that I had explained before. Another thing that we're looking at this time on ongoing work is looking at the localized displacements across the entire bundle. So as we mentioned earlier, we have the ability to map out how displacements occur on the bundle in the different regions. And we hypothesize that there will be differences across the whole bundle because our bundle is rather large. And then we really observed that there were no differences across the displacement on the overall mean. You can see here multiple regions of interest, branded here by different colors. And as the muscle bundle moves, even though we appear to see differences across the regions, in reality, whether we average by rows or average by columns, there are really no significant differences in the myobundle signal. Now, in summary, I hope that I have conveyed today that our young athletic stimulation myobundles appear to have higher dominant frequency and also higher mean displacements of the older sedentary. As you can see here, in terms of the mean displacement alone, it's basically 10 times, four times higher just by applying twice stimulation to the once daily stimulation. However, when we combine the tomatidine treatment again with the twice stimulation efforts, we appear to improve contractility of the older sedentary cells actually to degrees that are fairly closer to the young athletic, as we can see here. So we're roughly around the same values that we were observing for young athletic before any tomatidine treatment was added. So this is really encouraging data for us, and we are looking at hopefully replicating these results. And we will be updating you folks as we know more. Lastly, I would like to wrap up with indicating what how this actual model of muscle atrophy can be used in as an aging simulation model. We did develop this model for a spaceflight compatible. We actually had we flew it back in December 2020. Unfortunately, for technical difficulties, we weren't really able to have multiple replicates. And so the only thing that I can show you here is as we were able to observe in space how stimulation causes young athletic cells to contract the same way that we observe in the ground. So all of the data that's shown before were ground data, whereas this data right here is from actual in the International Space Station videos. Now we're looking forward to fly next week in another rocket launch. We're looking at having this system. I apologize. So we're looking at having this flight happening next week to hopefully getting a prolonged time frame and multiple replicas of this data. And in September or later in the fall this year, we're going to look at not only applying electrical stimulation studies, but also doing the drug delivery system that we just explained. And with this, I will just like to thank you everyone for listening, and I'll happy to take questions.
Thank you. It's wonderful. I'm a muscle biologist by training, so it's very intriguing to me. So have you looked at innervation for motor neurons to look at not just the sarcopenic or reduced cross-sectional area or atrophy in that, but also motor recruitment?
Truly not. Our system hasn't been developed to look at the interface of neurons and basically the neuromuscular junction interface. It is an interest of mine personally to look at that in the future, but with this collaboration that we have, we're focusing entirely on just the skeletal muscle parameters alone. But it is a very interesting area to pursue.
And then just one other. Have you looked at different exercise paradigms to look at, because I think the model would be really nice for looking at fiber type toxicity, right? And looking at like, say, a HIIT training, sort of like your young adult versus like a chronic low frequency stimulation, like endurance walking, and modulating that fiber phenotype and how drug toxicity differs between them.
Our group has not looked at multiple different doses of electrical or of the exercising regimes, but it is certainly of interest to us it is kind of future work or future aims to really look at multiple different, not only exercise regimes, but also different drug treatments. So it is in the works. We're currently looking at ways to characterize that, but we do have a limited, with the milestone-driven grant that we are under, we do have a very specific set of aims to accomplish.
Yeah. Anyway, I'll catch you after this. I've got quite a few more, but I'll let him get.
Hi, super informative talk. I'm definitely going to be eating a lot more green tomatoes. But as my question is, how long do you take these cultures out for with this repetitive testing?
We take them out for 18 to 20 days the longest. We don't go past that time frame usually.
So how does that in vitro timeline correlate to in vivo?
That is a great question. We have actually devised we have focused our timeline to be more in line with what we want to do in microgravity. So in reality, the in-ground studies that I just showed you were designed to be comparable to what we're doing in space. It is of interest to see how both co-cultures of different cell types and also extension of the culture time has an effect in the actual contractility parameters. We haven't looked at that yet, though.
Gotcha. Thanks. Good luck with your launch.
Thank you.
So I got a question. So it looks like you weren't actually monitoring the movement of the posts like most people do. You're actually looking at the fibers themselves, which is interesting. And it looked like in the young samples that you had a more uniform motion capture, whereas in the older individuals, you had intense areas and then areas with nothing. Did you see the areas that had intense with the older samples getting larger with time, or their intensity going up? And did you see increased fusion, or did you see more of an upregulation of the contractile apparatus of the myoblasts or the myotubes that were actually contracting?
I'm going to take that question by parts. So the first part of the question, I would say, we do not look at. So we did look at multiple regions of the pixel intensity changes, right? And we did see what appeared to be localized differences across multiple samples. But when we compare statistically, there were no differences between older sedentary and young athletic patients in general, as far as the region differences go. The other part was about.
Do you think that You think that you're getting increased fusion areas for recruitment in terms of it, or do you think that you are actually getting upregulation of the contractile apparatus and making the myotubes that were actually contracting stronger, or do you think that you were actually creating additional fusion around those areas that were contracting in the sedentary?
That is a great question. We have not looked at temporal differences because we take these videos, essentially the way that we take this is at the end of the culture period. So we do not track this over time, unfortunately. At the end of the culture period, then we look at how the muscles respond to the stimulation. And it is actually, I did not cover this, we do apply stimulation daily over, I think it's an hour that we apply it to. But now, these simulation videos that I showed you were just 40-second snapshots. So there might be some differences between how this muscle is responding over time that we are not capturing, unfortunately. There is in the works in the Malini lab looking at having a camera system that we can track the muscle bundles with time. So that it is currently in the works. We do think, based on the gene expression data that we have observed, that there seems to be differences between the older sedentary and the young athletic patients, and that the stimulation does appear to have some effect. So we suspect that it's more on the gene expression side.
One quick question.
Yeah, actually, this question is related to what Jay just mentioned. It's kind of, I'm not sure whether it is a recruitment issue or it is the maximum contraction issue. So yeah, I think you partly answered that question. Another question is, I don't know whether you have tested like a frequency dependent summation of the muscle. Because when you increase the stimulation frequency, the muscle has some summation and it goes up, climb up the tension. Have you ever tried that? Have you ever found anything?
We have not tried doing the summation to reach up to the titanic contraction that you're describing. We have not tried that. At the highest we did try was a 15 hertz frequency, and we did see that it appears to add up, but we didn't have enough samples to replicate that at the time. What I would say though is our camera system that we use to track the videos of these motions, they're not as high frame rates. So even if we apply a higher than 4 hertz frequency, we're not going to be able to capture movements within that range.